Yes, I've started my video.
Okay. Thank you. Thank you, sir.
Good evening, all.
Good evening.
A very warm welcome to all present here today. My name is Soumya De, and I feel immensely proud and grateful to introduce myself as the president of MBSI Swami Shraddhanand College. First and foremost, I would also like to express my deepest respect and gratitude to Professor A.M. Deshmukh sir, our esteemed national president of MBSI. Sir's wisdom, guidance, and invaluable contributions have continuously motivated and inspired not only us but everyone associated with the organization. His vision and dedication serve as a guiding light for all of us. I also extend my sincere gratitude to our respected mentor, Professor A. Archana ma'am, and the entire committee for placing their trust in me and entrusting me with this prestigious responsibility. Being elected as president of MBSI is not merely a position for me, but a heartful commitment to serve, lead, and contribute meaningfully to the growth of our society. MBSI has always been a platform that nurtures scientific curiosity, teamwork, and leadership. As a president, my vision is to strengthen collaboration among members, encourage innovation, and create opportunities that support both academic and professional growth. I strongly believe that true leadership is rooted in unity, responsibility, and collective effort towards a shared goal. Lastly, I would like to humbly acknowledge that today's inauguration has been possible with the constant guidance and support of our seniors, Alfez Bhaiya and Brahmjit Bhaiya, who are just not only our seniors but also ex president of this MBSI unit of our college. Their encouragement, experience, and mentorship have played a crucial role in shaping this event and guiding us at every step. With dedication and collective effort, I am confident that we will achieve new milestones together. Once again, thank you for believing in me. I assure you that I will carry out my responsibility with sincerity, honesty, and integrity. Also, everyone, we have an excitement announcement to share regarding significant milestone we have achieved as a unit. This official announcement will be made at the end of the event, so we request you all to stay tuned for this special moment. Moving ahead, it is a profound honor and privilege to introduce Professor A.M. Deshmukh Sir, the National President of MBSI, a visionary leader whose wisdom illuminates mind and whose guidance inspires generations. Sir's distinguished personality reflects a rare blend of intellectual depth, humility, and unwavering commitment to the advancement of scientific thought and academic excellence. His journey stands as a testament to perseverance, integrity, and selfless service, making him a source of inspiration for students and scholars across the nation. We feel truly grateful for the opportunity to learn from his experience and be guided by his invaluable presence. Over to you, sir.
Thank you. Thank you very much. I'm very happy to know that the Department of Microbiology of Swami Shraddhananda College, University of Delhi, is organizing a lecture series that is called as a scientific talk. The first topic is a gut microbiome, which is a very, very important topic. The maximum research in India and outside the India is carried out on gut microbiome. Simultaneously, there is an inauguration of MBSI unit. And I know that Shraddhananda College is a very, very active in Delhi. And the credit goes, of course, to the team, but the pioneer worker is a Dr. Archana ma'am. And at present, Dr. Shashi Chawla ma'am is a president of MBSI for Delhi. I really appreciate the efforts of Som Soumya Day, Sumayo Day for organizing this event, and his team, that is the vice president, secretary, treasurer, and other heads are there. Collectively, I am sure they will do the maximum best thing to percolate the knowledge of microbiology, and the maximum activity will be performed in the future. I know that Dr. Nishant Rai is going to speak in detail on the topic, so I don't want to take much time. My duty as a president is that I wish all the best for this event, and I hope that Dr. Nishant Rai will focus the light on gut microbiome. Thank you. Thank you again.
Thank you, sir. It was really a wonderful and thought-provoking talk or message for all of us and all of us unit. Your address was deeply inspiring and enriched us with clarity, perspective, and motivation. We are truly grateful for the opportunity we got and also the opportunity to learn from your experience and guidance. Moving ahead, it is my privilege to invite our respected State President of MBSI, Professor Shashi Chavla ma'am, who has been contributing in this esteemed position for the past 2 years. She has made remarkable contributions to MBSI Delhi State through her dedicated leadership, continuous guidance, vision, and unwavering commitment to academic and professional growth. She has organized the Lifetime Devotion Award function twice in Delhi. Her efforts have significantly strengthened the activities of MBSI. With great respect, we now request ma'am to share a few words on this occasion. Over to you, ma'am.
First of all, is my video? Am I audible?
Yes, ma'am.
Yes, you are. But your
Okay.
video is off.
Yeah, video is somehow not working. I don't
No
know.
problem.
There's some technical issue, but I'll speak. It's okay.
Okay, sure.
Later on, I'll join from my cell phone.
Okay.
Yeah. All right. I hope now it works. I don't know. Let me see. So, respected Professor Garg, who is principal of Swami Shraddhanand College, Professor Deshmukh sir, हमारे अपने MBSI के president, national president, Professor Archana, who is mentor for SSNC MBSI unit, and our esteemed speaker, Dr. Nitish Rai, my colleagues who will be here attending this talk, and my dear students. It's really nice. Every year I see Professor Archana. She is taking special efforts to make an MBSI unit, and you know that way actually it promotes these subjects, not only promotes the subject but also encourages students, because in various capacities you also get so many lateral skills. You know how to speak, you know how to make presentations, then how to write things. So many things come along with such events, such opportunities. So students become, it's like an holistic development, and definitely you all are learning microbiology. So it's really, I congratulate, first of all, Professor Archana, and then all of you who are here taking special efforts, doing events related to microbiology and promoting the subject of microbiology. So this is going to be an inaugural lecture, I believe, because I read on the poster it is inauguration of the unit as well. So I congratulate all of you. I see Sumayo, Devanshi, Priyanshu, whosoever is the office bearers. I mean, I don't want to read all that list on the poster, but we can read them and appreciate your efforts. So many congratulations for starting it. And the topic you are starting with, gut microbiome, that is my favorite subject. I think these days it is very in thing, I will say, in your language. So lot of people understand gut microbiome. They've started understanding. Or sir ne to shuru hi kiya hai with the that छाछ पियो, probiotic drink लो. So gut microbiome improve करने का तरीका है. So actually once we start knowing about it, we can always change our lifestyles and have a better health and immunity. So, once again, I'll not take much time. Thank you for inviting me, and congratulations for making the student unit, and also congratulations and my best wishes for having this lecture. So thank you so much.
Thank you, Shashi ma'am, for your inspiring and thoughtful words. Your address reflected deep commitment, clarity of vision, and genuine care for the growth of MBSI and its members. We are truly grateful for your continued guidance and leadership, which motivates us to strive for excellence. It was an honor to listen you on this occasion. Moving forward, it's my honor to invite a truly inspiring and selfless mentor, Professor A. Archana ma'am. She has always gone beyond expectations to guide us on the right path and nurture us into becoming a better version of ourselves. With her constant encouragement, compassionate mentorship, and unwavering support, she has shaped our academic journey in the most meaningful way. A true ocean of knowledge and wisdom, her presence itself is a source of motivation for all of us. We warmly invite Archana ma'am to share her valuable words with us on this occasion.
Thank you so much, Somyo, and very, very happy to see my students uh, organizing this session with utmost professionalism. And I really appreciate the hard work that you have all put in. So, heartiest welcome to all of you. Very, very good evening and many, many blessings for all. I thank heartily my dynamic principal, our college dynamic principal, Professor Praveen Garg sir, our mentor of MBSI, from whom I have learnt a lot, Professor Deshmukh sir, who is actually unstoppable and he catalyzes all good things. Whatever we want to do, he's always present and he never lets anyone down. We learn a lot of stuff from him all the time, all good stuff. And even his wife, Dr. Jyotsna ma'am, always encourages us, who's also retired HOD zoology. Then I really appreciate Dr. Sanjeev Patankar, who showed interest while he is in the train, but he has joined and his connection is not stable. But as and when I find that he can speak, we will definitely unmute him for a couple of minutes to take his blessings. It means a lot. Very, very happy to have my colleague and friend, Professor Shashi Chawla, who has very systematically and meticulously led the entire Delhi state for MBSI. And she actually, in a row, twice she has been the president, and very graciously and efficiently, she has organized many, many functions. And she spares her time to even come whenever we call her, and really grateful to her. Sir Louis Pasteur, who is known as the father of modern microbiology, said, "Chance favors the prepared minds." This is indeed true for my worthy students. I'm very, very proud of my students. It gives me goosebumps to see how they manage and tirelessly they have worked and they have proved themselves. I'm finding them, this batch, this the current lot generation Z and millennial generation, to be better than us, and this is the true evolution. So very proud that they have, on their own initiative, they have formed the MBSI SSNC student unit. Not only that, one can go on Instagram and check MBSI SSNC. They always discuss in detail microbe of the month, which is gradually educating even a layman. So this month organism is Akkermansia, which is a human gut microbiome organism. To match this, they have organized a talk by Dr. Nitish Rai, who is a shining star, and he is also my alumnus and the super senior of the current students. And I just indicated to him, and on a short notice, he was more than willing to deliver this talk. And you would be amazed. I appeal to you to hear him out completely because he's an amazing speaker. He makes all the tough things very, very easy to understand. I would just say, in short, what is the importance of being in a society, forming a society? So, when we make a society, we coexist, we learn, we impart, then we participate, we lead sometimes, and we follow the lead. We adapt with each other, we do teamwork, and we enhance our tolerance because we sometimes disagree and we agree to disagree. We have respect for other person's views. We have an open head, open heart. So, very proud that I have got the opportunity, along with my departmental colleagues who are very supportive, to form a society, not only MBSI SSNC, but also another departmental society, Sukshma, which another bunch of worthy students are handling, and they are in coordination. So each one gets their role, each one learns, each one teaches each other. And very special mention is of our three previous student teams, of whom I will just name the leads. The first and foremost was Mr. Brahmjeet, who is doing his PhD at ICGB Delhi. The second one is Mr. Abhikash, who is at NCBS. And the third one is Mr. Alfez, who is continuing with us in the fourth year and doing very meaningful research. And the fourth one is Mr. Somyo, who is in front of us, and their teams. So even if I don't name all the students for obvious reasons, it will be very lengthy. All of you are actually represented by these names, these four names. So these are we are very happy that even a person who is given a small responsibility, they listen to the lead and they are in coordination. Even if they make mistakes, they are open for correction and they do understand. And very great news is this year, this particular team has gone an extra mile, and they have left their comfort zone, and they have gone out of their way for starting a LinkedIn page of MBSI SSNC DU, in which all the updates will be there and all the hot research areas, scintillating research areas will be there. It will be actually doing good to one and all. So all are invited to follow the page and contribute, and we are all open for suggestions. It's their amateur attempt. They are not experts, but they have consulted many people. They have consulted the earlier presidents, and we had a online meet. And I'm very happy to see that they dare to dare, and this is what makes them who they are. Mentoring is indeed very enjoyable if the mentees respond well and they are full of gratitude. And I can't count my blessings in these terms. I'm very lucky as a mentor because I really enjoy mentoring, that I get to give them because they receive so well. They receive and soak it like a sponge. Even if I scold them, criticize them, if I am harsh with them sometimes, they have full faith that it's for their benefit. And I thank God for that, and I thank them also for that. So I'm really fortunate. I extend my best wishes and blessings to all students and my gratitude to all my colleagues. Some teachers from other departments of our college and even my own department are here, and some of the microbiology teachers from other colleges are here. We are very grateful to them for sparing their precious time to attend this meeting and share our joy. We are very happy today, and this all this to see this, I am really happy from the core of my heart, and this is the utmost fructification of my students' hard work. Thank you, and thank you for the opportunity given to me to speak here. Over to you, Soumya.
Thank you, ma'am, for your insightful and inspiring words. And actually, we are very fortunate to have you as a mentor. And your words were really motivating, inspiring, and will surely come across a good thing in our life under your blessings and guidance. We are truly grateful for the wisdom you shared and the encouragement you have always extended to us. Your guidance continues to motivate us to strive for excellence, growth, both academically and personally, as ma'am has mentioned, the holistical development. We deeply appreciate your time, support, and invaluable mentorship. Once again, thank you, ma'am. Moving ahead, now it's the time for the esteemed speaker. I would like to invite Navneet Pandey to give an introduction of our esteemed speaker, Dr. Nitish Rai, who's just not only our esteemed speaker, but also our super senior. Over to you, Navneet.
Thank you, Soumya. Good evening, everyone. Today, we feel extremely proud to welcome our super senior and an inspiring scientist, Dr. Nitish Rai, currently serving as an assistant professor in Department of Zoology at the University of Lucknow. He leads the Biogerontology and Neurobiology Laboratory, where his research focuses on aging, neurodegeneration, and herbal therapeutics. Dr. Rai completed his PhD from AIIMS, New Delhi, after topping his BSc from the University of Delhi and MSc from PG IMER Chandigarh, reflecting his outstanding academic excellence. Is the principal investigator of 5 major funded research projects worth nearly 1.72 crore, supported by reputed agencies such as UGC, Department of Health Research, and MHRD Rusha. With 28 research publications, international book chapters with Springer and Wiley, and impactful work on Alzheimer's and Parkinson's disease. His contribution continue to shape modern biomedical research. Dr. Rai has received prestigious recognitions, including the Young Scientist Award, MSI, Newton Bhabha PhD Placement Award, and NAB's Best Research Paper Award 2025. He is also a recipient of elite fellowship like Inspire, CSIR UGC JRF, and ICMR JRF, and qualified GATE Life Sciences with a 98.25 percentile. Today, we feel extremely proud to have him with us as our super senior and mentor figure, whose journey motivates us to dream bigger and work harder. Let us welcome Dr. Nitish Rai with a huge round of applause. Over to you, sir.
Thank you, Navneet. Thank you so much. Thank you for your kind words. And I sincerely don't feel like such a great personality when I sit in front of Professor A. Archana ma'am, my guru, my teacher, and I I feel like the same student what I was when at the time when I met her in at the SSNC college, and basically it's a privilege for me to speak in front of her and obviously the dignitaries. Professor Garg sir and Professor Deshmukh sir, who I have met once during my when I was there in the Sukhadia University, Udaipur, and it was a wonderful meeting. I can remember that very well, and also I'm very much thankful to my to Professor Shashi Chawla ma'am also, who is a state president of MBSI, and obviously, my gratitude to Dr. Archana ma'am for providing me this wonderful stage once again, and she basically she never forgets. Whenever she get a chance to invite me, she does so, and I'm so happy to interact with you all. And basically, what I can offer you all, obviously. It is actually my privilege to speak to you, to take this stage and to speak with you all to discuss this wonderful, basically, aspects of microbes. Uh, Basically, to all the ma'am, can I start the lecture if you allow?
Yes, sure. We are waiting.
Ma'am, just excuse me for a minute, please.
Yeah, sure. Meanwhile, can I request when you see it, our house full, some of the SSNC student, please make space for others. So I'm admitting 5 more are waiting. I'm admitting.
Yes. So,
Just
shall I
sorry,
share the
Nitish.
slides, ma'am? Shall I
Yeah,
share the
Nitish.
slides?
Yeah, Dr. Nitish. Dr. Patankar is here.
Right,
So,
ma'am.
just let him wish you and all of
Oh,
us.
right, ma'am. Right, ma'am.
Yeah,
Right, ma'am. Thank
just
you
a
so
minute,
much.
just a minute.
Please, please, please.
Yeah, can you find Dr. Patankar? And Patankar sir, are you there? I'm trying to find. Yeah, co-host, please help me. Dr. Patankar there?
I don't think I have searched.
Okay,
No,
sir.
ma'am.
Okay, sir. Okay, sir.
Patankar sir, please. But Patankar sir is
Hello.
not there. Hello. Yes, Patankar sir.
Here he is.
Yes, he's speaking from the train. I'll just add him at the spotlight. So Patankar sir is our MBSI national coordinator. He has been, and he has started under the with the suggestion of Deshmukh sir. He started the microbiology calendar. He has done lot of work from my MBSI to Ayush Ministry. Patankar sir, can you just speak a few words to bless all of us? I think his network is not stable.
It's okay, ma'am. We can wait. If you
No, no,
can
we can.
call, if you wish to call him, we can wait. Not a problem.
Hello.
Yes, he is there.
Yes, sir. We
Yes,
can hear
yes.
you, sir. We can hear
Yes,
you, sir.
Patankar sir, please. We can hear you. Patankar sir, please speak.
Yeah. Am I audible?
Yes, yes.
Yes, yes, yes.
Hello.
Yes, sir.
Yes,
Please
you are
speak.
audible. You are audible.
Yeah, yeah. So I am at uh, Chhatrapati Shivaji Maharaj Terminus, Mumbai. I wish you all the best, all the students joining this MBSI platform uh, for future planning and all other type of the thing. Uh, they must take the advantage of this particular forum and uh, utilize this for their future as such. So all the best. Thank you for joining me, allowing to join me. As I am on the station, so it is not possible for me to be here for more time.
Thank
Okay,
you.
bye bye.
Thank you, sir. Have a great, nice journey, safe journey.
Yeah,
We connect again. Thank you for
sure,
blessing
sure.
all in the meet. Thank
Thank
you.
you for the blessing, sir. Thank you.
Thank you. Yeah. Dr. Nitish, now you can start. You're rich with blessings.
Thank you so much, ma'am. Thank you. So I'm sharing my screen now.
Yes, please.
I must disclose that I'm not very well at this point. And if you hear some coughing in between the presentation, don't take it as a part of the presentation.
So we'll suggest to you to work on your microbiome.
Yes, ma'am. I have.
See, okay, please make it full screen.
Is my slide visible?
Yeah, visible. Make it full screen.
Yes, ma'am. Yes, ma'am. Okay. I hope I'm audible as well, ma'am.
Yes, yes.
Okay, okay. Thank you so much. Thank you. Thank you. So, dear all, I'll be presenting just a minute. Okay. So I'll be presenting this wonderful area of gut microbiota. And basically, this topic has been suggested to me by Dr. Archana, and I have this disclosure to make, that this is not an area in which I work, but I truly am. I have the microbiology is my first love, and I have taken my first degree in microbiology. So definitely, this is the one area which I have, which I read very interestingly, and it is basically quite an attractive area to which you all can work, and you all can pursue working in your future studies. You can definitely take up this area, and I'll tell you why, and I'll basically try to get your interest in this area. So so basically, what I'll be discussing today with you all is about the gut microbiota, and what all kind of the pathophysiological access it has. It has in the context of functional interaction, and what all therapeutic opportunities do we have in this area. So around 400 BC, one of the Greek physician, I'm sure you all must have heard about the Hippocrates. He's called as the father of medicine. He has that understanding that death sits in the bowel, and the bad digestion is the root of all evil. So you see, you try to understand, try to comprehend that human beings have that idea about the gut from the time when there was so much little known about the human body as per se. So that, that has been given that kind of importance from the time immemorial. So what is basically a gut microbiota? What do we understand by the gut microflora? So basically, it is the huge community of these tiny microorganisms that basically live in your digestive tract. And basically, which area of the digestive tract is basically your intestine, right? So I think day before yesterday, I was surfing, I was browsing my LinkedIn, and I got this very thoughtful picture where it was written that whenever you feel lonely, just remember that there are millions of bacteria living in your body. And since that your body is the home to those microorganisms, you mean the world to them. So basically, you can think how much deep this saying can go. Right, because we know that these gut microbiota, or per se, you can say the human microflora, they are there when you take, like when a baby is born. The gut microbiota takes starts to reside within the body, right? So obviously there are reports that there are tiny microorganisms, or that there are tiny microbial DNA that has been seen in the fetal microbes as well. But then the major community of microbes that come in the body immediately after the birth, when the baby is exposed to the basically exposed to the microbial environment, then it basically the whole microbiota resides in the basically in the characteristic part of the body, right? So you can comprehend that how much important these microbes are to us, and how much important are we to them, right? So human gut microbiome is basically its area is is expanding very rapidly, with particularly active work going on in the mechanistic area, like the what are the interaction between the microbiota and the host, what are the disease focused access, and what are the development we are getting in on the context of the therapeutics, right? So if if microbiome is so important, obviously we can use them, utilize them to treat a certain disease, right? We can use them to basically suit their need for a particular human being, so as to give the area of personalized medicine. If you can think, if you can comprehend what I'm talking about, is basically you can see that there are there are areas being developed now where they are using these microbes to cater them for a particular human need, for a particular patient need. So that comes under the area of precision medicine approach, right? So basically, much of these microbiome research field is basically transitioning, is going under a phase of transition where they are not mere descriptive associations with the body, but they are being seen as a causal, translational, and for the therapeutic approaches. So obviously, it is being treated as an essential organ of the human body, because you see, we are as much bacteria as humans. One, now, why I am saying this? We are as much bacteria as humans, because you see, there are uh, lots and lots of microorganisms residing not only in the gut but in the other areas of the body as well. So if you take the ratio of the bacteria to the human cell, you will realize that it is approximately 1:1, right? So that is why I'm saying we are about as much bacteria as we are humans. Obviously, these groups of bacteria, archaea and eukarya, which are colonizing our gastrointestinal tract, we call them gut microbiota. Okay. So I think we are clear about the gut microbiota. We are clear about the human microflora, which is obviously a major umbrella term under which the gut microbiota comes, right? Now, what is there in the gut microbiota? So if we talk about the what all bacterial species, what all microbial species resides in the gut, they are characterized by several phyla, which includes your Bacteroidetes, Firmicutes, then other minor groups like Proteobacteria, Actinobacteria, uh, Verrucomicrobia, Fusobacteria, Cyanobacteria. So they are present. Obviously, these are basically the minor. But again, don't think them as a minor, so they are not important. No, like your Akkermansia, it comes under your Verrucomicrobia, right? And we already know, as Professor Archana ma'am also told me a few days back, that this bacteria, we have taken it as the microbe of the month. So obviously, its role has been seen in diabetes, type 2 diabetes. It is being currently being used as a therapeutic, right? We are using it to treat the several diseases, right? So if they are so much important, obviously, we cannot undermine their role, because simply because they are less in number. Their number is less because it is in the perfect combination of the human gut microbiota. Its number is ought to be less, right? With the proportion of other major phyla which are there, its number is less. But then its role is not at all minor. So it is basically a simple representation of how your gut microbiota, they are there in the body, in the gut, obviously. So there are large portions of firmicutes, bacteroidetes, and then there are others which comes under your 5%. So this is basically and now again, this could not be true for a child who has been just born, right? It guts microflora would be different, right? Because gut microflora would differ depending on the stage in which the individual is, right? So this is basically about, if you can see, if you could comprehend it, you would characterize this as a gut microbiota composition of any adult, right? So how does this gut microbiota, it basically resides in your gut and not any other pathogenic bacteria? For example, like E. coli or something like Enterobacteriaceae family, or any other pathogenic strain. Why not? Why only these gut microbiotas? Why they are there? So obviously, as I already told you, that when a child is born, not much is known. When you are in the fetal stage, not much is known about the gut microflora, but definitely that field is again open to research. And right now we cannot say much about it, but again, from the moment we are born, we are come to this world, through the process of labor and childbirth, the gut microflora comes into existence, right? The major after few months, the baby feeds on the natural milk or on the formula milk, how frequent the baby is exposed to the microbe, what geographical location is the baby surviving. So all these things comes into picture when we talk about the gut microbiota. Is there any question? I'm sorry. If you all don't have any question, please kindly mute your mic, please. And you can definitely stop me anytime, and we can discuss things. If you want to ask me anything, if I would know the answer, I would definitely would like to share some insights, or we have several professors with us who can guide us. But again, if you don't have any question, please try to mute your mic, please. Okay. So why does this gut microbiota does not allow the pathogenic organisms to survive? Because they are having the immense capacity to outcompete these pathogens. Now, how do they do it? How do they do it? You can see here are the gut microbiota. Okay, I'll take you to these complex pictures. Don't get I mean, don't get confused by seeing so many data in the picture. Just focus on what I'm trying to tell you here. So you can see the gut microbiota, right? They release certain factors, like they release nutrients, they release these bacteriocins, they release these SCFAs. What does these SCFAs means? These are short chain fatty acids, right? So these metabolites, what do they do? Bacteriocins, they directly kill these pathogenic organisms, right? Bacteriocins, they kill these microorganisms by inhibiting their cell wall synthesis, by inhibiting their protein synthesis, and basically by degrading their DNA. They are degrading their their rRNAs, tRNAs. Okay, but how does this SCFA work? They basically inhibit the viral gene expressions. They can decrease the expressions of these genes which are present in the pathogenic bacteria. You can see the gut microbiota also basically enhances the inflammatory and immunological mediators of these B cells, Th17 cells, and macrophages, right? So they basically do not let these pathogens stay in the same niche, and they have a tendency to compete for the nutritional niche and physical niche which is there present at the dispense of these bacteria. So they do not get to have their share of nutrition in the same area where these gut microbiota are. Okay, so so this is how our gut microbiota doesn't let the pathogens take over their place in the habitat where they are there. Okay, but again, when we take these antibiotics, when we take the over-counter medicines, it affect the gut microflora, right? Not only that, our diet, our lifestyle, our area, how we maintain our body, all of it affects the gut microbiota. Okay, we'll talk about it. We'll talk about it. So, what do we understand about gut microbiota when in the context of human pathophysiology? So, we know that there are lots and lots of gut microbiomes which are now being seen as a causal factor for certain pathophysiology of obesity, hypertension, cardiovascular diseases, diabetes, cancer. Inflammatory diseases, mental diseases. So it all has been these microbes, they have been seen as a major progressor of these processes. Obviously, in my previous lecture, when I delivered lecture around I guess around 3 4 years back, I think I told you about, I we discussed about the development of major human diseases and the gut microbiota. Obviously, your batch was not there at that time. But then I discussed these factors, these diseases with you all about the gut microbiomes and how they basically they have an effect on these diseases, right? But again, today I'll be not be discussing about these diseases, but rather about how does these gut microbiomes have so much control over these diseases. Why it is that we are saying that okay, hypertension is happening, it is because of the gut microbiome. Okay, because we are getting cancer, it is because of the gut microbiome. Why it is why you should take it seriously and not as a something as a fluke? No, because what we have something is called as the gut axis, right? Gut pathophysiological axis. Okay, we'll be discussing it in more detail in our coming slides. First of all, you should understand what is dysbiosis. So basically, dysbiosis is nothing but a state of altered gut bacterial composition. Okay, gut bacterial composition. So that means when your gut microbiota, it has to have in a certain proportion. So I'm sorry. So for example, if bacteroidetes and firmicutes have to be in the major proportion, like proteobacteria and actinomyces, if the other bacteria have to be in a lower composition. Now, if this state gets altered, if more and more pathogenic bacteria or those bacteria who are not the normal habitats of our gut, if they appear in the gut, then this state is called as dysbiosis. Simple as that. Okay. So कोई भी बैक्टीरिया जो हमारे गट में नहीं होना चाहिए, अगर वो वहां पर है, तो that means this stage is called as dysbiosis. Okay. Okay, so why does this dysbiosis happens? It can obviously our gut microbiome can be affected by major factors, as I already told you. You can see in this figure, your gut microbiome could depend on the infant feeding method, like how the baby is getting fed. Is the baby getting fed by the mother milk or by the infant formula milk? That will definitely will impact the gut microbiome. Then what is the birthing process? Was the birth natural, or was the birth about or the delivery was through cesarean process? Then at what life cycle stage you are at? You are a infant, you are a adolescent, you are an adult, you are a old age, mid age. What all life stages are you in? That will again will impact the gut microbiome. Then what geographical location are you there? Like again, you can see that there are lot and lots of less number of loose dust you will experience in the western part of the world, in the UK or in those countries in the Europe. You'll see less and less dust, right? So definitely the microbial exposure is also less. So that will again impact the gut microbiome. Or like, are you a frequent user of over-the-counter medicines? Are you a frequent user of antibiotics? Uh, what is your diet? How much balanced diet you are having? How much fast foods you are having? Or how much pro-inflammatory diet you are having? Uh, there are studies which have been shown that gut microbiota has been very much largely favored by the Mediterranean diet. That is a kind of a healthy diet. Okay, so there are studies, you can get lot of studies about these diets and impact on gut microbiome. Then how do you cope with this stress? Do you do exercise? How much metabolic stress you have? How much psychological stress you have? You don't feel about the psychological stress as a problem now, but believe me, give and take 10 years, 15 years, you will start feeling the effect of stress psychology as well. मैं क्लियर हो रहा हूँ? आप लोगों को समझ में आ रहा है चीजें, या मतलब मैं बहुत ज्यादा डिटेल में चीजें ऐसे बोल रहा हूँ जो नहीं समझ में आ रही है? प्लीज आप
It's very, very
जो भी आप लोग सुन
simplified.
रहे हैं, प्लीज आप लोग अपना प्लीज ओपिनियन दे सकते हैं. प्लीज, am I
Nitish,
making myself
yeah,
clear?
it is very, very simplified and very impactful, Dr. Nitish.
Thank you, ma'am. Thank you.
Yes, sir.
Okay. So as I already told you, we'll be discussing this time about the disease focused axis. So what do we understand about the axis word? Axis simply means that there is a connection, right? There is a connection. So you can see this is a gut. And you can see this is a brain, your nervous system, this is your immune system, and this is your metabolic system, right? So there are three axes, right? Well, first is the brain, gut brain axis. Then there is your gut immune axis, okay? And then there is your gut metabolism axis. Obviously, this is a liver, and liver is the master regulator of our metabolism, right? So what is this axis? In my previous slides, I told you that, okay, we are having cancer. Gut microbiota is the maybe the reason or one of the major contributor, not the only reason, obviously. We are having diabetes, gut microbiota. We are having immune dysfunction, inflammatory disease, so on and so forth. Everything is related to gut. Why are we saying that? Why are we saying that? Why not any other thing? It could be any other organ, right? Why only gut? Because gut is the only organ which is in constant communication with the three major organ or the three major system of our body. I'm so sorry, it is not an organ, but the system of our body. So what first is the gut brain axis. Second is your gut immune axis. And third is your gut metabolism axis. If you see your major organs, they have been covered under these three heads, right? Under these three systems, right? So, if we talk about the gut axis, it is the way of simply saying that your gut is connected with the other major systems in your body. And this system is not only unidirectional as shown in the arrow. No, it is bidirectional. If the gut is talking to the brain, brain is also talking to the gut. Okay, immune system is also talking to the gut. The liver or the other metabolism associated organs, they are also talking to the gut. Okay, so this communication is basically a bidirectional communication. Okay, so that is why this because of this communication, the gut and the gut microbiota becomes so important. And why are we saying this? Is why we are saying that, okay, gut microbiota may be the cause and may be the key to therapeutically modulate the disease associated with all these three systems. Okay, so this is the major highlight. This you should understand this thing first. Okay, so coming on to the first axis, which is your gut brain axis, which simply means that there are certain microbes in your gut, and obviously they are talking to the brain, and they are having in a constant they are in the constant communication. The brain and the gut, they are in the constant communication by the several mediators, right? So this is a simple picture that has been made by the use of AI, and it has been beautifully summarized the core concept what I wanted to convey, so I used it. But obviously this has not been made by me, so there you can basically you can find some mistakes, but again I've tried my best not to keep any mistakes. So anyway, so so basically these brain and gut, they are basically having the communication using the immune signals, using the hormones, and using the mediators, molecular mediators, which are basically involved in this type of communication. Okay, so we know that how our gut and brain axis is having a bidirectional communication. We have established it, right? So there are microbes which are present, and now these microbes which are residing in the gut, they are releasing several molecules, several molecular mediators in the gut, obviously, and they are going to the brain, right? And they are going to the brain, and they are basically reshaping the brain development. They are reshaping our response to a certain stress. They are reshaping the cognition, which is a major function of our brain, obviously, and they may have a role in our neurodevelopmental process, right? Obviously, if they are having role in their in our cognition, then you can easily comprehend that they might have role in the cases where there is no cognition or there is reduced impaired cognition, like the neurodegenerative disease, like the Alzheimer's or the Parkinson, right? The like now this has not been said by a fluke, but obviously there have been carefully planned experiments where they have showed that there are instances of altered neurogenesis, altered myelination, and altered behavior in the animal which has been made germ free. Now how can you make an animal germ free? By treating it with antibiotics, right? You can make them microbiota free, right? And if it is happening at a certain stage, then what effect does it cause? Now this all comes under the animal study. You take a mice and you treat it with antibiotics. Obviously, you can take different stages, life stages of the mice. You can take the mice at different life stages, and you can treat them with antibiotics, and you can study the effect, right? And obviously, its importance has been we have also observed it on in our daily life as well. We have observed it in the movies and whatnot. So how the brain and the gut, they are totally related with each other, right? So if you see what I've told about the germ-free mice, it's basically a mice where we treat them with antibiotics, and you can see the gut microbiota is basically finished, right? So there is no microbiota in the gut, so there is no gut-brain axis. Now, what does it? What kind of pathological implications can it have on the organism, right? So obviously with the normal mice, there is the process of neurogenesis. There is a process of dendritic growth. There is an intact blood brain barrier, which is so much important for keeping our brain for protecting our brain from the xenobiotics or the any other entities which are not supposed to enter in our brain. But then it gets disturbed when there is no gut microbiota. Then these blood brain barriers and these neuro process of neurogenesis, they get disturbed. Again, the process of myelination and the process of the synaptic plasticity, the microglia, they all get disturbed when we don't have these gut microbiota, right? Because their role is there in all these normal physiological processes. So there are several factors which you can see that there are factors like antibiotics, drugs, stress, diet, circadian rhythm, even how much sleep you are getting. These all factors, they affect the basically the your gut microbiota composition, and not only gut microbiota composition but also your brain behavior as well. So you can see how your blood brain barrier gets damaged when you are exposed to these factors. So there are blood brain barrier, there are blood cerebrospinal fluid barrier, then there are the choroid plexus vascular barrier. How they get disturbed. And when you see about the gut barrier, you can also see the disturbances in the GVB, which is basically your gut vascular barrier and gut epithelial barrier. Okay. There are certain microbiota which are whose composition get disturbed, and how these disturbed composition, they release the microbial metabolite, which are again not the same which is there in the normal which is not there in the normal microbiota composition. So again, if the microbiota composition will be normal, it will release certain kind of metabolites, but if they are disturbed, they will not be releasing those metabolites but certain other metabolites. Now these certain other metabolites will definitely disturb the blood brain barrier. Okay, so this is how the gut brain barrier, and this is just one of the process. Okay, because I didn't have to go into much detail, so I have tried to give you only a glimpse of what goes wrong when we talk about the gut brain axis. So this is, if you want to read more about this, you can go and read this paper. This is a wonderful paper from the Albert and Cryan 2024. It has been published. So basically, you can see that there are two barriers which are getting disturbed. First is the blood-brain barrier, and second is the gut barrier. The blood-brain barrier and the gut barrier both get disturbed. Okay. So now this inter-barrier communication is also disturbed because the microbes is getting disturbed, and therefore the metabolite which they are releasing is again getting disturbed. Okay. So coming on to the gut immune and inflammatory disease axis. So what is gut immune axis? Is basically is the close partnership between the microbes which are present in your intestine, the gut wall, and the body defense cells. That means those cells which are there residing in the gut area, the microbes which are there, and basically their interaction with the immune cells, right? So again, you can see when the gut and the microbiological flora, they are balanced with the immune cells. It is the healthy scenario. And when there is dysbiosis, when no longer we can see any normal gut microbiota present in the gut, then basically they charge on these immune cells, and they elicit their response, and basically they causes the inflammation, right? So basically you can comprehend it in a way that when any gut microbe is there, and when its composition get disturbed, that means when there is a condition of dysbiosis, then what they do is basically they causes the initiation of something called as a leaky gut. Now what is leaky gut? Leaky gut, as I already told you, that when there is a process of dysbiosis, what happens is your gut barrier gets disturbed, right? Gut barrier gets disturbed. Now, if there is dysbiosis, normal microflora is not there anymore. That means no longer there will be a normal metabolite. Now, if these abnormal metabolites will be released from these abnormal gut microbiota, they will be basically be causing certain pathological process, one of which is chronic immune activation, right? Now, this chronic immune activation, what it is basically when our gut microbiota elicits the inflammatory response in our immune cells, right? And causes them to elicit more and more of the immune response. Now, this is the cause of several diseases which are obviously inflammation associated. One of the major disease is the inflammatory bowel disease, or also called as Crohn's disease. Now, remember one thing, that this inflammatory bowel disease or the Crohn's disease is basically one of the first diseases where it has been clearly established that there is a definite role of gut microbiota in this disease. Okay, so I would encourage you all to read more and more about it, because it has been established beyond any doubt that IFD is basically caused due to the dysbiosis and the presence of the pathogenic gut microflora. Okay, so again, there are certain other inflammatory diseases which could be seen as basically as a result of the process of dysbiosis. And what happens is dysbiosis, there is a loss of the natural diversity, and basically disruption of the uh, gut barrier. What is gut barrier now? Obviously, gut barrier is where these your cells, epithelial cells, they are basically tightly wrapped with the tight junctions and the mucus, right? Now, when this epithelial cell junction, it gets loosened, it gets leaky, then it causes the microbes to throw their microbial products into the body. And now what does this microbial metabolite will do? It will launch the chronic immune activation, right? So I hope I am making myself clear. With this figure, you will be understanding it more and more. So you can see that there are certain microbes, gut microbes. Now what is happening, if in here, if you can see, there are short chain fatty acids, and there are certain metabolites like butyrate, right? Now butyrate is one of the microbial metabolite which has been studied quite well in the body. And you see butyrate has a role in activating GPCRs, which have a role in reducing the inflammation, right? Now, suppose if there is no longer there is bacteria which can produce this butyrate, what will happen? It will disturb the GPCRs, will activate the inflammation, right? Will activate the inflammation. Now, why does this happen? Why does bacteria no longer produce butyrate? Because no longer there is normal microflora present. Because now there is Enterobacteriaceae. No longer there is Bifidobacteriaceae. No longer firmicutes. Okay. Now, who will produce butyrate? No one. What will happen? Inflammation will happen. Right. So this is just the one of the mechanisms which you can see here. How does this how does this affect the normal process which is undergoing in any human being, right? So obviously it also affects the beta oxidation. It also affects the low oxygen which is present inside the lumen of the intestine. So again, if there is if there is no longer low oxygen is there, then obviously the gut microbiota will be disturbed, obviously. Okay. So once again, you can see that this is the characteristic of the healthy gut microflora. So you can see how the symbiotic gut microbiota, they are releasing the bacteriocins, they are releasing the metabolites. And here, when there is dysbiosis, their metabolites are changing. Their metabolites are changing, and what it is causing? It is causing the immune activation, pro-inflammatory response, right? Now, this pro-inflammatory response, what it is causing? It is causing the gut barrier disintegration, right? Now, this gut barrier disintegration, what is happening? The metabolite which are being released from these pathogenic bacteria, they are being transferred into the bloodstream. Now, where they are going? They are going to the different body organs. So now these body organs, they will be affected by these microbial metabolites. And this effect will not be in good cause, obviously. So therefore, because of these characteristics, we are so much convinced that gut microflora is the root cause of several diseases, right? So there are altered neutrophil, macrophages, dendritic cells, and innate lymphocytes, T and B cells, which drive the cytokine production. And obviously, it is the root cause of why there is this tissue damage, which is there in the inflammatory bowel disease. And basically, there are pro-inflammatory responses. This has been seen in several diseases because of the several metabolites, which I have already told you about, right? Now, coming on to the third and the most important gut metabolism axis, which is basically nothing but the network, but the connection of the gut microbiota with the several organs, including your liver, which are basically concerned with the energy production, right? So how much energy do we take from the food? How much energy we need to spend on certain process? It all occurs. It all happens because of the communication with the gut microbiota, right? Just excuse me. Okay. So so as I already told you, how much energy we can harvest from the food, how much how much we can take from the food we have just ingested, how much basically whole metabolic uh, milieu will be regulated by the gut microbiota. So you can see that there are instances of microbiota transfer from obese versus lean donors in the animals, and how they have improved the insulin sensitivity, for example, in any animal. So there are studies where they have done that. And again, as I already told you, how much sleep you are having will definitely affect the your metabolism. And why? Because of, again, this circadian disruption is also altered by the microbial flora present in your gut, right? So basically, it contributes to the lipid and energy imbalance. Then again, gut microbiota, it communicates with the liver, muscles, adipose tissues, and several other peripheral organs via the metabolites and the immune signals, which form the multi-organ metabolic axis. This is very important because, again, as I already told you, that these metabolites, they do what? They basically they communicate with the several organs. Now we know how these gut microbiota, they are so much relevant in the disease associated with those organs which have nothing to do with the gut, right? Because why? Because these gut microbiota, they are releasing their mediators into the bloodstream, which are going and interacting with those peripheral organs, right? Okay. So how does this gut metabolism access comes into picture? Because again, we know that these gut microbiota, they utilize several metabolites, several substrates in the body, and they produce certain products, right? Now these products, they are having tremendous effect on the body, particularly on the molecules, on the proteins, which are the master players in the metabolism, right? Like you can see, there is a concept of hyperglycemia, there is a concept of glucose uptake, there is a concept of immune signaling. How does these process, these like significant process in the body, they get affected by these several products which are being formed by these gut microbiota. Like TMAO is a major metabolite produced by the gut microbiota. Then there is enterolactone, cresol sulfate. What are these molecules? PERK, AMPK, ERK. These are the proteins which are absolutely important for the conduction of these process. Okay, AMPK is a kinase. PERK is again a very important molecule, signaling molecule present in the FOXO signaling. These FOXO signaling and insulin signaling, they are very much important for these metabolic processes. Now, these proteins, they get affected by these different metabolites which are being released from the gut microbiomes. Now, if these gut microbiomes will not release these mediators, then definitely these processes will be affected, and therefore your metabolism will be affected. Okay, so it is as simple as that, and as easy to comprehend that why does these microbiota, they have the role. Okay, now coming on to the therapeutic implications, there is a concept of fecal microbiota transplantation, which is also called as FMT. They are being tested very vigorously in several of the diseases, including your IBS, MS, and multi-drug resistant infections, right? Then there are engineered microbes and bacteriophages which are being developed selectively for treating certain diseases like the depression and certain neurological disorders as well. Now, again, there is a trend of precision and personalized medicine. If certain human is undergoing a number of pathologies, now what kind of best microbial composition we can give to this particular patient so that their whole body comes under like comes to a certain phase where we could basically bring the body back to normalcy. Okay, again, uh, using only microbes may not work, but using these microbes along with the existing drugs might do wonders, right? So uh, there is this more and more trend of these personalized microbiome medicine which is coming into existence. Okay, which is changing the treatment paradigm like anything. And then there are certain, obviously, there are lots and lots of product which are present in the market nowadays for you, which are using these microbes as the treatment and as the nutritional supplements for basically bringing the body back into the equilibrium. And what could be the future directions? Again, there are lots and lots of work which has to be done, and definitely lots and lots of discoveries which has to be made in the field of gut microbiome and in the field of gut microbiome area. And obviously, the very little is known till now about these gut microbiomes, and this area has not been very old. So this is very recent area, and many of the labs in the West are working like anything on this area. And obviously, uh, telling you a very practical aspect, working in this area is not also a very much difficult and doesn't demand that kind of money as well. So you can definitely start these work in India, even in India, and you can do wonders. It is basically a very good area of study which you can undertake. And obviously, this area is moving towards the translational end. So getting the fundings from the Indian government and from the like private parties also is not is not that difficult. Okay, so the ability to tailor these therapies according to the individual personal microbiome composition will definitely be a very good idea, and basically it will definitely add to the personalized medicine approach. And I would request all of you to go and read more and more about this. So I hopefully I have made my point very much clear, and hope I gave you all something to take back and go and read about it. I hope so. So is there any question? I would be willing to take it.
Thank you,
Thank
sir,
you.
for delivering such an insightful and enriching session, especially on the simpler terms like dysbiosis, gut microbiota axis, and gut-brain connection. Uh, Personally, it's my favorite topic as well, so I actually enjoyed it, and it was explained very specifically and very in a simpler manner. Your explanation of how environmental and host factors influence disease focused access, how the microbiome regulates our physiology, and your introduction to concepts like leaky gut have truly broadened our understanding of human health. More than just information, your talk has created genuine curiosity among us and opened new ways of thinking about the invisible yet powerful world within us. You have helped us to connect complex science with real life relevance and making the subject both fascinating and meaningful. The figures, the flowcharts, and the visual representations you used it here made complex mechanisms much simpler and clearer for us understanding. You have not only shared an advanced scientific knowledge, but also created genuine curiosity and excitement to explore this field further, especially the fields like precision personalized microbe medicines, engineered microbes, and bacteriophages, and also the fecal microbiota transplantation. We are deeply grateful for your time and your clarity and the passion with which you shared your knowledge. Thank you once again, sir, for inspiring us to explore deeper, question more, and pursue science with greater enthusiasm and purpose. I would request the curious minds out here, if you have any questions, you can type it in the chat box of the Zoom meeting room.
Not only questions, if you have any personal insight, what all you want to share, anything. आई नो उस स्टेज पे इतने क्वेश्चंस होते नहीं है हमारे पास। अगर कुछ क्वेश्चंस हैं भी तो हम काफी ज्यादा किसी भी गेस्ट के सामने बोलने में उसमें काफी ज्यादा हमें मतलब एक तरीके से एक, महसूस होता है कि हम नहीं बोल पाएंगे या नहीं बोलना चाहिए वॉट एवर। बट अगर कुछ भी आपको शेयर करना है, कुछ भी आपको लगता है, कुछ भी आपने सीखा या समझा, या कुछ भी आपको खुद का पर्सनल शेयर एक्सपीरियंस से कुछ शेयर करना है, यू आर मोस्ट वेलकम बच्चा। ऑल ऑफ यू, ऑल ऑफ यू। ऐसा कुछ
I
भी नहीं
want
है कि
to
यहाँ पर कोई बहुत ही मैम ने खुद ही इतना लाइट कर रखा है, एंड देयर इज नथिंग लाइक दैट, है ना। लोग प्लीज
So
ओपन
I
होकर के
want to
जो भी शेयर
say,
करना
Dr.
चाहते हैं।
Nitish, I want to say आजकल के बच्चे वैसे नहीं जैसे तुमने कहा. They are Gen Z and they are millennials and they are very active and they some of them have studied microbiome and I have taught and along with me another alumnus very bright, Dr. Vikram Kumar has taught and he is a champion of microbiome. So he's also in the audience. So we can also invite him to share some more points if he wants. I think some questions are coming. So I want to say it was a houseful performance. You can say you can see the number of participants. More than these, there are more on the live stream YouTube. So they
राइट
have
मैम।
enjoyed and learned.
Truly blessed, ma'am, truly blessed. And as I've already told you, I'm not an expert, but I'm still a learner of this wonderful field. And obviously काफी ज्यादा इंटरेस्टिंग है मेरे खुद के लिए भी ये सब जानना, नई चीजें सीखना अपने फील्ड से हट करके। मेरा एक्चुअली कोर एरिया एल्जाइमर्स है, जिसमें मैं बायोमार्कर की स्टडी करता हूँ और हर्बल थेरेपेटिक्स की में इन्वॉल्व हूँ। बट अगेन ये एरिया ऐसा है जिसका कि ऐसा नहीं है बिल्कुल भी कि बिल्कुल अलग है, मेरे फील्ड से कनेक्टेड है, डेफिनेटली बहुत कनेक्टेड है, और डेफिनेटली एक तरीके का मेरा लगाव है माइक्रोबायोलॉजी के साथ, हमेशा रहेगा।
Some questions are there in the chat box.
Yes, sir.
Soumya, yeah, who will take the
I want to
questions?
read the
Yes,
questions.
Please,
yes.
please, please.
There is a question from Manvi Negi. Sir, how can the birth processes like C-section and natural birth affects the gut microbes?
Very good question. So आप एक चीज खुद सोचो, अगर कोई चाइल्ड बर्थ है, if you compare the two process, cesarean versus natural birthing process, you can see the exposure, microbial exposure, जो content, जो basically जो vigor है, जो force है, जो basically एक आपका environment है, totally वो change हो जाता है. अगर आप process की बात करो, एक cesarean की और या फिर एक natural birthing process की, जो भी आपके microbes हैं, जो भी आपके environment है, it completely changes, right? सो अगेन ये एक चीज अगर आप एक मेरे ख्याल में एक पेपर था बाय नियाजी एट ऑल, it was published in 2023. If you all can go and read it, it's a wonderful paper about how the birthing process can affect the microbial composition occurring in a baby. Okay, तो बेसिकली उसमें उन्होंने यही बात बताई थी that the birthing process, in what the exposure, the microbial exposure, it changes during the two types of birthing process. So definitely it affects how the microbial composition developed in a child, right? Again, how that baby is getting fed, what is the, like, is the baby being fed only on with the mother milk, or the baby is getting fed with the formula milk, only with the formula milk, or with both? The 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 microbial composition will be totally different. The geographical area in which the baby is living, again, will change the microbial composition, totally different, right? So all these factors comes into play when when basically we talk about the gut microbiota. So again, you all have to go and read more and more about it. This is not the definite knowledge, but definitely this is where somewhere you can start, right?
Thank you, sir. Sir, there are some more questions. There's a question from Pranay Kumar Khan. Sir, would we consider the microbes who populate our mouth as a part of gut microbiota, as well as they are, I suspect, surely going to affect our health? And how are we going to make it all personalized for, say, one patient, especially in the context of fecal transplant?
The gut gut itself means, strictly term में अगर बात करूं तो no, obviously intestine area में जो microbes हैं, we can call them as a gut microflora, right? फीकल जो ट्रांसप्लांट की बात हम लोग करते हैं, एफटीएम की बात हम लोग जो करते हैं, उसमें बेसिकली अगर आप देखो तो एक स्टडी थी, आई थिंक पार्किंसन के केस में एक स्टडी आई थी, जिसमें कि उन्होंने क्या किया था कि एक नॉर्मल माइस से उन्होंने बेसिकली उसके उसके फीकल मैटर से उन्होंने माइक्रोब्स को एनरिच किया था, एंड देन दे ट्रांसप्लांटेड इट टू अ डिजीज्ड माइस, एंड what they have seen is that there was a reduction in the death of the dopaminergic neurons. तो वो ये सारे जो एविडेंसेस थे, ये सब अर्ली एविडेंसेस थे एफटीएम के, फीकल ट्रांसप्लांट के, कि कैसे आप माइक्रोब्स को ट्रांसप्लांट करके एक हेल्दी हेल्दी इंडिविजुअल से एक माइक्रोब्स को ट्रांसप्लांट करके हाउ यू कैन ट्रीट अ सर्टेन कंडीशन. दीज वर द अर्ली एविडेंसेस, राइट? अब इसी को अगर मतलब एक्सटेंड किया जाए टुवर्ड्स द अदर डिजीजेस, तो डेफिनेटली इट कुड बी वन ऑफ द अप्रोच through which you can start or attempt to basically get a therapeutic meiotic, right? Therapeutic एक तरीके से आपको एक मिल जाता है एक perspective, right? So again, this could not be seen as a solitary approach, no. Obviously you have to check all the boxes, you have to satisfy all the other mediators, you have to take considerations of the other approaches as well, and then this combinatorial approach may be more effective for treating a certain condition.
Thank you, sir, for your explanation. There is one more question from Kartik Rajpal. Sir, except maintaining healthy lifestyle, is there any other suspectable way to maintain healthy gut microbiota?
Obviously, having a good diet doesn't only checks all the boxes. You have to deal with the psychological stress as well. अगर कल अर्चना मैम ने टेस्ट अनाउंस कर दिया तो वो एक स्ट्रेस है, आपको साइकोलॉजिकल स्ट्रेस है जो आपको डील करना पड़ेगा, राइट? आपको कल मैम ने डांट दिया, आप बदमाशी कर रहे थे क्लास में, कुछ आपको मैम ने डांट दिया, तो हाउ यू टेक इट, राइट? एंड ऑब्वियसली आपके फ्रेंड्स हैं, आपकी फैमिली है, सबके साथ आपका इंटरेक्शन है, सबके साथ आपकी एक बातचीत है। अगेन, हाउ यू डील विद देम, how do you take your life, how do you maintain your life, how what kind of routine do you have, right? Circadian बात मैंने आपसे कही थी अभी, स्लीप साइकिल की बात कही थी, राइट? स्लीप साइकिल आपकी कितनी ज्यादा रेगुलेटेड है, ये सारी एवरीथिंग कम्स इनटू पिक्चर, राइट? एवरीथिंग कम्स इनटू पिक्चर, बहुत ही क्लियर है, ये सारे मैसेजेस बिल्कुल क्लियर है, इसमें कोई डाउट नहीं है। दिस इज वेल एस्टेब्लिश्ड, बच्चा वेल एस्टेब्लिश्ड है कि ये सारी चीजें आपके गट माइक्रोबायोम को अफेक्ट करती हैं। एंड गट माइक्रोबायोम एक ऐसी चीज है जो सबके साथ इंटरेक्ट कर रही है, इसीलिए इट ऑल कम्स इनटू पिक्चर, राइट? सो ये सारी जो मैं चीजें आपको ऑलरेडी आपके स्लाइड्स प्रेजेंटेशन में आपको ऑलरेडी ये चीजें बताई हुई हैं, दैट इज द वे। ऑब्वियसली कहना बहुत आसान है, करना बहुत मुश्किल है, ऑब्वियसली, बट अगेन दैट इज व्हाट वी आर हियर फॉर, टू लर्न एंड टू ग्रो, राइट? भी मैं जैसा था बीएससी में भी वैसा नहीं हूँ, थोड़ा ग्रो किया हूँ, शायद थोड़ा और ग्रो करूंगा, but again you all have to grow in that way.
Thank you, sir. The next question here is from Mayank Chauhan. How do gut microbes influence brain function and behavior through gut-brain axis, and what implication does this
Sorry,
have,
sorry, sorry.
especially
Again,
for
please, can you repeat the question? Sorry, sorry. Your voice got
sure,
cut.
sir, sure. How do the gut microbes influence brain function and behavior through the gut-brain axis, and what implication does this have for the mental health conditions?
ये जैसा मैंने आपको बताया ना, कि जितने भी न्यूरो डेवलपमेंटल प्रोसेस हैं, ऑल योर न्यूरो डेवलपमेंटल प्रोसेस, माइलिनेशन हो गया, न्यूरोजेनेसिस हो गई, डेंड्राइटिक फॉर्मेशंस हो गया, जो भी आपके मेन फंक्शंस चल रहे हैं, मेमोरी, कॉग्निशन, everything is related to the metabolite what these gut microbiomes are releasing, right? So as I've already told you that these gut microbiota, they are feeding on certain substrates and they are producing certain metabolites which are very much important for regulating the important molecular mediators, important signaling molecules. Now, if these mediators, if these metabolites will not be there, they will be indirectly be affecting the molecular signaling pathways, signaling molecules, which will affect the pathways, which will affect the process. अभी आप देखो डिप्रेशन में क्लियर रोल दिख रहा है, डिप्रेशन में क्लियर कट रोल आ रहा है गट माइक्रोबायोटा का। अभी आप देखो पार्किंसन में क्लियर कट रोल आ रहा है, अल्जाइमर्स में क्लियर कट रोल आ रहा है, ठीक है? सो एसेंशियल ट्रेमर में क्लियर कट रोल आ रहा है। तो बेसिकली इतनी सारी डिजीज के साथ गट माइक्रोबायोटा का रोल देखा जा रहा है। तो व्हाई इट इज अकरिंग, बिकॉज ये बेसिक बायोलॉजी को अफेक्ट कर रहा है, आपके ब्रेन के बेसिक बायोलॉजी को अफेक्ट कर रहा है, ठीक है? टू वे डायरेक्शनल जो बाई डायरेक्शनल इंटरेक्शन चल रहा है आपके गट और ब्रेन के साथ, इट इज दैट इम्पॉर्टेंट, वो इतना इंपॉर्टेंट है। और इसी वजह से अगेन, अगर आपका गट बैरियर डिस्टर्ब्ड है, डिसरप्टेड है, अगर आपका ब्लड ब्रेन बैरियर डिसरप्टेड है, इट विल अगेन डिस्टर्ब द बाई डायरेक्शनल कम्युनिकेशन, और ये आपको प्रॉब्लम करेगा, फिर नेचुरली ये प्रॉब्लम करेगा, कॉज करेगा, राइट? इसीलिए लीकी गट देखा जाता है, इसीलिए लीकी ब्लड ब्रेन बैरियर देखा जाता है आपके इन डिजीजेस में। ये नॉर्मल है, नेचुरल है। आप देखोगे तो आप कई सारे पेपर्स आपको मिल जाएंगे जिसमें ये सारी चीजें रिपोर्टेड है, ऑलरेडी रिपोर्टेड, इट हैज बीन वेल एस्टैब्लिश्ड बियॉन्ड एनी डाउट।
Yes, sir, definitely. So there is a question how aging can affect our microbiome.
आप लोग बहुत नॉर्मल से नेचुरल चेंजेस हैं आपकी बॉडी में होते हैं, विच यू विल रियलाइज जैसे जैसे आपकी बॉडी एज होती है। ऑब्वियसली वी ऑल आर एजिंग, राइट? राइट फ्रॉम द मोमेंट वी टेक द बर्थ वी स्टार्ट टू एज, राइट? सो अगर आप देखो तो बेसिक माइक्रोबायोलॉजिकल कंपोजिशन जो गट माइक्रोफ्लोरा है, दैट itself changes with the aging process, right? अगर आप देखो तो कुछ मैंने लास्ट जब मैंने प्रेजेंटेशन हुई थी मेरी इस पे गट माइक्रोफ्लोरा पे, तो आई टोल्ड यू अबाउट सेवरल ऑफ द माइक्रोब्स which basically which are not there in the adult stage, but it occurs in the aged stages, right? So these again जो भी आपके स्ट्रेसर्स होते हैं, जो भी आपके क्रोनिक इन्फ्लेमेटरी मीडिएटर्स होते हैं, जैसे मैंने आपको ऑलरेडी बताया कि अगर आपके गट माइक्रोबायोम में जो भी मेटाबोलाइट्स हैं, if they are releasing into the body, they are obviously we are experiencing stress all over our life, and we are exposed to these definite factors which are again making our gut leaky, which are causing the microbial metabolites to flow through the body. And one of the major event which occurs is basically chronic inflammation, right? For example, I'm taking the example of chronic inflammation. There are lots and lots of example, but I am taking chronic inflammation. अब ये भी आप जान रहे हो कि आपका gut leak करेगा, आपके microbial metabolite will release some metabolites which will cause the chronic inflammation, right? So there is some level of chronic inflammation will definitely be there in the body. Now what does this chronic inflammation will do? It is one of the mediator of aging, right? It is one of the mediator of aging. So there are lots and lots of explanation to this question. ऐसा नहीं है कोई एक चीज मैं बोल दूं वो सत्य है, बट अगेन एजिंग ऐसा प्रोसेस है जिसको अभी तक डिफाइन नहीं किया जा सकता. एजिंग का मल्टीपल कॉजेस कहा जाता है. जैसे आप कहा जाता है कि रिडॉक्स इम्बैलेंस है, मतलब एंटीऑक्सीडेंट और ऑक्सीडेंट्स का इम्बैलेंस causes aging. Then there is chronic inflammation causes aging. देन टीलोमियर शॉर्टनिंग कॉजेज एजिंग. तो बहुत सारे ऐसे फैक्टर्स है जो एजिंग कॉज करते हैं. तो एजिंग इतना अपने आप में कॉम्प्लेक्स. बट देन अगेन इफ आई हैव टू एक्सप्लेन हाउ द गट माइक्रोफ्लोरा कैन कॉज एजिंग, दिस इज वन ऑफ द explanations, right? One of the explanation could be this, right? तो एजिंग अपने आप में कोई ऐसी चीज नहीं है जो कि आप एक बार में समझ जाओ. आपको पता होना चाहिए एजिंग में सिर्फ हाइपोथेसिस है, कोई डेफिनेट आंसर नहीं है एजिंग का. आज तक एजिंग का कोई डेफिनेट आंसर नहीं है ना? तो इसीलिए एजिंग एसोसिएटेड डिजीज का भी कोई जवाब नहीं हमारे पास है क्यों होता है. बट अगेन होता है, वो सब हम सब देखते हैं, राइट? सो अगेन एजिंग में आपको स्ट्रेस से कोप करने की सिचुएशंस कम, आपके पास ताकत कम होती है, वही फ्रेलिटी कहलाती है, राइट? फ्रेलिटी इज व्हाट? फ्रेलिटी इज सम इज अ डिसऑर्डर वेयर वी हैव द लेस कैपेसिटी टू कोप विद द स्ट्रेस. अगर हमें फॉल का इंसिडेंस हो गया तो हम दोबारा उठ नहीं पाते. हम उठ पाएंगे लेकिन हम उस तरीके के नहीं बन पाएंगे जैसे हम पहले थे. क्यों ऐसा हुआ? एजिंग की वजह से, राइट? तो फ्रेलिटी कंडीशन क्या है? एजिंग एसोसिएटेड कंडीशन है, राइट? तो एजिंग एक ऐसा टॉपिक है जिसको आप कोई इसका डेफिनेट आंसर नहीं है अभी तक, है ना? कोई इसको एक्सप्लेन नहीं कर पाता. डेफिनेट हाइपोथेसिस है, उन हाइपोथेसिस के फॉर और अगेंस्ट में चीजें हैं सबके, हर एक हाइपोथेसिस के.
Yes, sir, definitely. Sir, one last question. Sir, how do microbial metabolites interact with drugs?
ये आप कह सकते हो एक तरीके से कि माइक्रोबियल मेटाबोलाइट may or may not interact, but definitely microbes will interact with the drug, right? If we are suppose exposed to a certain drug, uh, it can impact the growth, it can impact the uh, whole metabolism of the commensal gut microbe which is there, right? Which is there residing in the gut. And therefore if like for example antibiotics are very well known examples, right? How they can flush off all the gut microbiomes from the body. Definitely there might be some instances where the microbial metabolite might also be interacting with the drug, but then again I don't think this could be a very major factor, because again metabolite is being continuously produced by the microbe, right? If that metabolite might be interacting synergistically or antagonistically with the drug, doesn't matter. There will be certain numbers of metabolites still being produced by the microbe, right? But the drug will be harmful or be good in respect to the gut microbiome which is already there in the body, right? So if that drug is causing the harm to the gut microbiome, then it's a big problem rather than if it is interacting with the metabolite. Okay, because again if that metabolite is antagonistically working with the drug, you can obviously increase the dose of the drug, right? You can modulate the drug dosage. But again, if that drug is causing the loss of the gut microbiome, then it's a problem, because then again you are losing the source of those metabolites, which are very much important for the body, right? वो क्वेश्चन ज्यादा इंपॉर्टेंट है कि आपका ड्रग जो ले रहे हो आप, वो आपके माइक्रोबायोम को तो डिस्टर्ब नहीं कर रहा है। वो क्वेश्चन ज्यादा इंपॉर्टेंट है रेदर देन मेटाबोलाइट के साथ कोई एक्ट कर रहा है कि नहीं कर रहा है। ठीक है ना?
Thank you, sir, for
ये
answering
आपके
all the
प्री क्लिनिकल
doubts.
स्टडीज में यही तो देखा जाता है। प्री क्लिनिकल स्टडीज में यही तो देखा जाता है। क्लिनिकल ट्रायल्स में यही तो देखा जाता है कि वो ड्रग का जो है वो मतलब हमारे किसी ऐसे मेटाबोलाइट के साथ तो नहीं एक्ट कर रहा है जिससे कि कोई प्रॉब्लम आएगी, मतलब उस ड्रग का कंपोजिशन चेंज हो जाए, या उस मतलब कंपोजिशन पर से मतलब मेटाबोलाइट चेंज हो जाए, अंदर जाकर ये कोई ऐसा मेटाबोलाइट तो नहीं कॉज कर दे रहा है, कोई फॉर्म कर दे रहा है। कहने का मतलब ये है, राइट?
Sir, there were some more questions. Like, how can anyone know if they have a leaky gut or not? Before onset of any disease related to microbiota, if you are able to recognize leaky gut, can the disease be prevented? And also, sir, have any particular food which is present that increases the gut microbiota?
हाँ, प्रोबायोटिक्स। हम लोग प्रोबायोटिक की बात करते हैं इतनी सारी, आपका माइक्रोब ऑफ द मंथ उसकी बात कर लो अगर आप, तो इज बीइंग यूज्ड टू ट्रीट द टाइप 2 डायबिटीज। अभी उसका बहुत ट्रेंडी चल रहा है और बहुत काम हो रहा है इस बेसिकली इस बैक्टीरिया पे। So obviously they are being used, and I mean, there are lots and lots of researchers who are working in this field. Probiotics का फील्ड पूरा ओपन है इस चीज के लिए। और अब इसी के लिए तो बात मैं आपको बोल रहा था कि इसीलिए तो आपका ये जो पर्सनलाइज मेडिसिन की बातें हो रही हैं, हाउ वी कैन टेलर द माइक्रोबियल कंपोजिशन पर से फॉर ट्रीटिंग अ सर्टेन टाइप ऑफ डिजीज, वो इसी की बात तो मैं कर रहा हूँ, इसी चीज को तो मैं बोल रहा हूँ। तो डेफिनेटली इस चीज को चल रहा है, ये चीज चल रही है, राइट?
Thank you, sir, for catering all the curious minds here, and thank you for explaining in the most simpler manner we can get it. And sir, once again, thanking you for the lecture. It was really, really beneficial and insightful for us. And now we shall move. We shall move to the next part of the event, that is the actual inaugural of the unit of this tenure, that is 25-26. And I would take immense pride to invite our Vice President, Mr. Priyanshu Chanchal, to introduce himself and share a few words and take the mic.
Thank you so much, Soumya. Moving forward, good evening, everyone. I feel extremely honored to present before you today.
There's
My
a
name
network glitch.
is Priyanshu Chanchal, a third year introduce myself as Vice President of MBA to express my sincere gratitude. Am I audible now?
Yes, please continue.
Yes, you are audible. You are audible, Priyansh. Go on.
Please continue, Priyanshu. Could someone else volunteer to substitute? Because he's
Yes,
facing a
ma'am, I'm
problem.
continuing.
First of all, I
Yes,
would
ma'am.
like to express my sincere gratitude to our respect trusting me with this important responsibility. Part of MBSI is not just a position for me.
Priyanshu, are you trying to speak? I think he is not able to get the network. So,
Thank you, Priyanshu, for your wonderful message. Now I'm moving to the next member, our secretary, Mr. Navneet Pandey.
Thank you, Soumya. In science, progress happens not by working alone, but by collaborating, questioning, and building together. Good evening, everyone. I'm Navneet Pandey, a third-year student from Department of Microbiology, Swami Shraddhanand College. Feel truly honored to be here today as General Secretary of MBSI. I sincerely thank our respected mentor, Archana ma'am, and the core team for placing their trust in me. This role is not just a position for me, but a responsibility, a commitment to serve with dedication, integrity, and teamwork. Just like how different cells and systems work together to sustain life, MBSI also thrives on collaboration and coordination. As General Secretary, I ensure smooth communication between teams, organize and manage society activities effectively, maintain discipline, transparency, and support every department so that we function as one strong, united body. With our mentors, constant guidance, and collective efforts, we can achieve remarkable milestones and create a lasting impact together. Once again, thank you for this wonderful opportunity. Looking forward to an inspiring journey ahead. Thank you.
Thank you, Navneet.
Thank you.
I want to invite our media head Kanaya to share his few insights.
Good evening, respected faculty members, esteemed chief guests, and dear friends. I am Kanya Kumar, a student of third year of Microbiology Department and the media head of MBSI. I am grateful for the opportunity to be part of this esteemed organization led by Achna ma'am. As media head, I am responsible for managing digital presence, including social media, event promotions, and creative content. My goal is to communicate scientific activities clearly and creatively so that our work reaches a wider audience and encourages student participation. Thank you.
Thank you, Kanaya. I would like to request Ms. Semal, who is our PR head, to share a few words.
Good evening, respected members, esteemed guests, and my dear friends. I am Semal, student of the third year from the Microbiology Department, serving as the Public Relations Head of the MBSI SSNC Delhi Student Unit. I sincerely thank our mentors and the core committee for their guidance and support. For me, this role is more than just a designation, it is a commitment to serve society with dedication and professionalism. I will work towards ensuring effective communication and meaningful outreach for our initiatives. And we strongly believe in collaboration. While each member has defined roles, we aim to work without rigid boundaries, supporting one another and contributing collectively to the success of our activities. And with your continued support, I look forward to contributing positively to the growth of our student unit. Thank you so much.
Thank you, Semal. I would like to ask our treasurer, Ms. Prachi, to share a few words.
Good evening, respected members, esteemed guests, and dear friends. I am Prachi of third year Department of Microbiology, and I'm very proud to serve as the treasurer of the MBSI SSNC Delhi Student Unit for this year. I'm deeply grateful to our mentor Archana ma'am for her constant guidance and support, which truly motivates all of us. Taking on this role, I'm highly passionate and committed to handling my responsibilities with honesty, dedication, and accountability, assuring the smooth functioning of our unit. I look forward to giving my best, contributing meaningfully to the growth of our student unit. Thank you.
Thank you, Prachi, for your experience and insights. Now I want to introduce our creative team behind this unit, that is our media team. And I want to start with Aram Ahmed.
Respected dignitaries, teachers, and friends, good evening. My name is Aram Ahmad, and I am a second year student in the Department of Microbiology, Swami Shraddhanand College. I feel truly honored and privileged to be part of this inaugural event today. It is a genuine pleasure for me to serve the MBSI SSNC Delhi student unit in the media team. I look forward to work under the guidance of my seniors, and I am eager to contribute to the success of our unit. As microbiologists, we spend our time studying the invisible. However, my duty in the media team is to make our work visible to the world through media. Thank you, everyone.
Thank you, Aram. I want the next person here, of media team is Miss Arzu Verma.
Good evening, everyone. Today, it is my honor to attend this meeting with respected guests and my teachers and my dear friends. I am Arzu Verma, a second year student of microbiology and a member of MBSI SSNC unit in the media team. I'm very thankful for the opportunity to work with such supportive seniors and classmates. I have learned many things in this team and I'm looking forward for more opportunities in the future. Thank you.
Thank you, Arzu. And now I'm moving to the last member of our media team, Harikesha.
Please unmute yourself.
Sorry, I want to invite Harikesha, the last member of our media team.
I'm sorry, actually. Good evening, respected members, esteemed guests, our teachers, and my dear friends. I am Harikesha, a second year student of the microbiology department and a member of the MBSI SSNC unit media team. I am sincerely grateful to our mentors, seniors, and the core committee for giving me this opportunity. Being part of the media team has been a valuable learning experience, helping me to improve my creativity, communication, and teamwork skills. MBSI is not just a society, but a platform that encourages overall growth and learning. I assure you that I will continue to contribute with dedication and sincerity towards the society's vision and activities. Thank you once again.
Thank you, Harikesha. Now moving towards our public relation team, the team which manages all the connections between different institutions. Starting with Saurav.
Science only creates impacts when it is communicated well, and that is where I came in. Good evening, everyone, respected teachers, my fellow members. My name is Saurav Rathor, and I'm proud to be a public relations member of the society MBSI. Microbiology is not just about microscopes and labs, it's about how science connect with society. As a PR member, my role is to give that science a voice, share our work, our events, and our ideas in a way that reaches and inspires people. I look forward to building strong connections, promoting meaningful initiatives, and representing MBSI with clarity, creativity, and responsibility. I'm excited to grow, learn, and contribute to making our society more visible and impactful. Thank you. I'm looking forward to an amazing journey with MBSI.
Thank you, Saurav. Now I want to invite Ms. Pallavi for us to share her insights.
Good evening, everyone. I'm Pallavi Kujur, a second year microbiology student. I'm honored to serve on the public relation terms for the Microbiology Society. As a PR member, my mission is to give our research a voice, representing MBSI with clarity, creativity, and professional responsibility. I look forward to enhance our society's visibility and make a meaningful impact together. Thank you.
Thank you, Pallavi. Now moving towards our event coordinator, starting with Devyanshi.
Hi, good evening, respected teachers, esteemed guests, faculty members, and my dear friends. My name is Devyanshi Tyagi, and I am a second year student as well as the event coordinator of Microbiology Society of India, Delhi unit. Being a part of MBSI truly means a lot to me. This role has given me the chance to explore my interest in microbiology beyond textbooks, while also helping me to grow in areas like leadership, coordination, and communication. Working alongside with such supportive peers and mentors has been both inspiring and rewarding. And I am genuinely grateful for the trust, guidance, and encouragement I have received along the way. Thank you for giving me this opportunity. I look forward to learning more, contributing wholeheartedly, and continuing to grow with MBSI.
Thank you, Devyanshi. Now we move towards our last event coordinator, Yukta.
Thank you, Bhaiya. Good evening, everyone. It is an honor and a privilege to be a part of MBSI. I am Yukta Vijay Raghavan, a second year microbiology student, currently serving as the event coordinator of MBSI SSNC unit. We are grateful for the presence of our esteemed guests, mentors, and participants whose support and engagement continue to inspire our efforts. Your participation truly adds value to today's program, and I sincerely hope that this event has proved to be informative, engaging, and beneficial to all of us. Thank you for being a part of this gathering. It was a very successful and enriching session by Nitesh sir. Thank you again.
Thank you, Yukta. As I see that our Vice President has been here, so I would like to invite him for his introduction.
Thank you so much, Soumya. Moving forward, good evening, everyone. I feel extremely honored to present before you today. My name is Priyanshu Chanchal, a third year microbiology student, and I am truly grateful to introduce myself as Vice President of MBSI Swami Shraddhanand College. First of all, I would like to express my sincere gratitude to our respected mentors, seniors, and the entire fellow members for trusting me with this important responsibility. Being a part of MBSI is not just a position for me. It is an opportunity to learn, grow, and contribute to the academic and professional development for our society. As the Vice President, I aim to work with dedication, teamwork, and transparency. I will always try my best to support my team and ensure that society continues to grow stronger through meaningful activities, scientific discussions, and collaborative efforts. We are trying to maintain and propagate legacy which made by our seniors and working closely with my fellow members. Once again, I sincerely thank you all for giving me this responsibility. I assure you that I will carry out my duties with full commitments and give my best for the progress of our society. Thank you so much for your time and support. Okay, now we are moving ahead. As Archana ma'am said, we are extremely proud and excited to announce that the official LinkedIn page of MBSI SSNC will be going live very soon. This is a major step and milestone forward in building a strong professional presence of our society and creating a platform that connects science students, opportunity, and innovations. Our LinkedIn community will showcase MBSI achievements, events, research activities, collaborations, and students' initiatives, while also helping members grow professionally through exposure, networking, and learning opportunities. This is not just a page, it is the beginning of a powerful digital identity for MBSI. Let's come together to make this platform active, impactful, and inspiring. Stay tuned, support it, share it, grow with it. MBSI SSNC is ready to make its mark. As we are nearing the conclusion of today's insightful event, I would now like to respectfully invite our esteemed and beloved mentor, Professor Archana ma'am, to kindly propose the vote of thanks. We would be honored to hear your concluding remarks as we formally express our gratitude to everyone who contributed to the success of this event. Over to you, ma'am.
Thank you, my very, very dear students. Very, very proud of you. You have put this up at a very short notice in a brilliant manner, and I couldn't ask for more. You have really exceeded my expectations. So, for the formal vote of thanks, actually, I'm very thankful to all present here who contributed in any direct or indirect manner, because due to all of you, the event has been a huge success, and a couple of hours which we have spent here together have actually enriched the entire system, the academia, and also you have made an outreach via the LinkedIn page of yours. First of all, worth $1 million appreciation is very enlightening and enriching talk by Dr. Nitish, who is our very, very celebrated student, and his way of explaining was like this from the since his first year. I must say this, he has had a glorious journey till now, making a difference. So the audience was spellbound, including me. Even if we know about microbiome, his way of presentation was very nice, very lucid explanations and lively slides. And as we could see, there were dozens of questions. The moderator, Somyo, could take only half of them. So we are thinking of having doubt solving session in-house in the college. Dr. Vikram is there, who is also junior, much junior of Dr. Nitish, and he is also a champion of microbiome. So I think I'll arrange that. Many thanks to Professor Deshmukh sir, who has been the engine of our fast train of progress. And Professor Sanjeev Patankar sir, who was so enthusiastic that from his journey also he was very eager to join. Professor Praveen Garg, obviously our principal sir, who is always motivating us to do good things and always backs us up in all such worthwhile endeavors. Professor Shashi Chawla, I am very, very grateful to you. She is a dear friend and a very wise faculty, and she has always given constant encouragement, and we are very thankful to her. She spent her precious time blessing our students. We are extremely grateful. Some joinees I could see in the the attendees. They were from national level, from outside Delhi, and some were unknown names also. It's very appreciable that they have quest for knowledge and they attended our meet to make it star-studded. The entire MBSI SSNC team, office bearers, they have done their roles very nicely, very brilliantly, and it was a flawless kind of, they put up a flawless kind of event. I want to specifically thank my departmental colleagues, Dr. Vikram, Dr. Vineet, and Dr. Rahul, for constantly being there and listening carefully and enjoying to be teachers when earlier they were students and they used to be doing events like you are doing today. So life has come a full circle for them. Still, they are here to encourage all of you and me also. Many thanks to them and many, many blessings. Then previous MBSI SSNC Delhi unit student teams led by Mohammad Alfez Sheikh, the just previous one, and Mr. Brahmjeet Dahiya, who was the opening batsman, the first and foremost president. Also Mr. Abhikash, who could not attend today, but these guys have spent their time prior to this event for enriching you, the present batches, by their rich experience. And I could see, because I was there in all the meetings, so I could see very clearly that they were teaching you many things which they have done and succeeded, and many things which they could not do, but they were telling you so that you evolve, you exceed their performances also. So all this, like, it is a first, second, third, fourth floor happening, but the plinth is very important, and the previous floor and its strength gives more and more strength to the upper floors. So I want to thank them wholeheartedly and bless them, because they have enriched, they have set up an example worth emulating, that this is the way to go, and we have to encourage the forthcoming generations to do even better than how we have done. Very, very important is to appreciate Mr. Prathmesh for providing us in a timely manner the Zoom platform, for having such immense patience. Mr. Prathmesh assists Deshmukh sir and MBSI for all these tech savvy activities, and he is a student himself, but he devotes time in the evenings for our esteemed society MBSI. Also, he is operating the live stream YouTube, and we can only send many thanks and gratitude and lots of best wishes for his bright future. Thank you, Prathmesh. Last but not the least, big thanks are due towards today's amazing audience from Delhi and outside, and we saw that the meet was very well attended. Now also I checked a few minutes back, many attendees are present in the YouTube live stream as well. And many thanks to my sweet students who graciously shifted from here to the live stream so that our guests can get entry into the Zoom meet, and this is what makes life go. not only academics, but these sensitivity, empathy, and help, mutual help, is extremely important. And actually, my heart is full of gratitude right now, and I bless all of you. We owe the success to all the audience. Many, many thanks. And we really hope that you gained from today's event. From the scintillating talk, about the talk, I want to add that not handling our body and our life wisely causes dysbiosis, and conversely, correcting the gut microbiome can restore whatever losses we have suffered due to our own negligence. And so it is the pain and it is the cure. So as we deteriorate, when we find it out, we can restore. So microbiome makes actually our lives rock, and I would urge and motivate everyone to read about microbiome. It's the amazing permanent solution of all our health problems, including physical, mental, emotional, all the conditions. So I'm extremely happy and very indebted to all of you, and heartiest congratulations to be the first one among the student unit nationwide to have the MBSI LinkedIn page where you can impart information, do good deeds, share and care, and give some of your glorious legacy for your descendants. Thank you. Very good evening. And I wish you a great evening ahead and enjoy the winter time, especially in Delhi. Many, many thanks. Namaste, namaste, and Jai Hind. Thank you.
Thank you, ma'am. Thank you so much. Thank you.
Thank you, ma'am.
Done, ma'am.
Thank you so much, ma'am.
Thank you. Pratmesh,
Yes, ma'am.
you switched off, right?
Yes, yes, yes.
Okay, thank you, Pratmesh. Can we continue only for 5 minutes here?
Yes, you can continue. Yes.
Okay, thank you, Pratmesh. God bless you. You are
Thank you.
awesome.